we had him on to discuss biofuel. I really loved that show. What did you think of that one?
Oh, I thought it was awesome. I learned that we could make diesel to run my truck out of slimy algae. And I'm wondering why the hell we're not doing that.
Well, now I'm hearing the biofuels being used like all over the place. But what was surprising to me was how effective. I just sort of figured biofuels would still be putting carbon into the environment. That was my understanding. It's just, we're not. It's not as bad. But what we learned about biofuels in that one was really shocking and hopeful. It's the first time I've had some hope for the environment in a while. Hoover Dam, as I understand it now, is not even producing any electricity now. The river there is so dried up. It's a little crazy, but we decided we wanted to have him back. He talked about nanotechnology, he's a microbiologist, and so we decided we wanted to have him on again to talk about just what it is that he does, and so he agreed, which is surprising. He was actually on the first time, and he still decided to come back for another show. So let's bring in now Greg. Greg, how are you?
Hello. How's it going?
It's going good. It's going good. I'm so glad you decided to come back. You were really interesting last time, and
thank you.
You had so much to talk about. I was just like, oh my God, we're just you're screwed. Now you whet the appetite. Now we got to bring it back. So tell us a bit about yourself.
Yeah, so I'm a PhD candidate in microbiology, and when I was going through the microbiology professors, they were really fun, interesting, like environmental microbiologists, looking at the microbes in the environment. There was some that were doing these, a really popular thing right now is to study how microbes in our gut are impacting our bodies and our mind and that kind of stuff. And that was really interesting. But then this one professor in plants actually pulled me into one of the interviews, and he was doing nanotechnology based stuff, and I had never even considered it. I looked him up, and I mean his papers were. It was like science fiction. It was absolutely incredible. Bomb sniffing plants. Light emitted, like plants that are emitting light, stuff that I'm just like, what? This is so cool. And I actually, so that's why I joined the lab, and I've been working on nanotechnology based approaches to study microbiology, ever since.
I do want to bring up one thing just because you brought it up, and I've heard this before. I guess there is a new theory that at least it's possible that some memories are stored in the gut. Is that accurate?
It's the first that I'm hearing about it. I know that the microbiome, our gut microbiome, plays a big role in our immune response. So a lot of that is, to deal with the production of, I forget if it's T or B cells. But it's a big thing to do with our immunity, and it has a big, there is some like gut microbiome changes, differentiations between like elite athletes and the public, which is, interesting. Why is that? What are the differences? If you took those differences and you put them in your gut, would that make you like an elite athlete? That kind of stuff. It's really interesting microbiology questions.
Excuse me. Damn. All right. Well, let's see the $5 super chat. Sister Porpoises, thank you so much for your support and for your super chat. We really appreciate it. Quote: "Technology is awesome, but computer science is better than biotechnology." Fighting words. Also, see my last joke about recursion. Really.
Yeah,
really,
that's cool. Cool. I mean, a lot of the computer science stuff is actually making its way into biology. So, a lot of people like personally, I kind of think as like chloroplasts in plants, like 3D printers, so we can kind of like make them. And there's actually really interesting. So there's a new field coming out. I mean, that's happening right now. It's called synthetic biology. And if you research biology, you kind of know that there's this biology black box. And the black box is everything is set up right. Everything is all mapped out. Everything should go right. But then it just fails, and we don't know why. And that's kind of the black box of biology. There could be you're trying to make a protein, but then there's like a protease that you don't know about that chews your specific protein down and takes that apart. So synthetic biology is trying to remove that black box, basically, so we can research and use biology for specific purposes and go into these in my case, microbiology, but go into those microbiology worlds and kind of like see what's there, see what proteins are being made. So there's actually complex logic gates that happen within synthetic biology. So like if this protein is present, then you make this protein, and then that makes this protein, or else this protein is made, and those are kind of like fluorescent proteins. So there's a technology called that's used a lot. It's called FRET, fluorescent resonance energy transfer. So a lot of the fluorescent proteins will emit a certain wavelength in the presence of just themselves, or another one entirely different if there's another protein there, and we can kind of like do if-else logic gates within microbes and organisms now. So it's really cool. It's not an if-else, and it's not two different things now. Computer science and biotechnology is definitely interdisciplinary.
That's incredibly fascinating. But what I really want to know is, when am I going to be able to sign up to get my SSD implant so that I can remember shit?
It's a good question. Yeah, I mean, there's Ray Kurzweil's like singularity, right? Where he talks about like you kind of like what was that?
No, please. I'll
I mean, that's the kind of like that's the real. I mean, I can think about and pontificate about maybe like future technologies that would do that, but I guess that's kind of like the whole like I'm uploading my brain into like something larger than myself kind of idea. It is kind of. I think that's not too far off, to be honest with you. A lot of the, specifically if we're talking about nanotechnology, there's a lot of nanotechnology approaches that augment current organisms, so that allow them to, nanotechnology approaches allow you to, monitor wavelengths. They allow you to, in the brain, they allow you to, in plants, they allow you to, see stress levels or the presence of certain particles that the plant has taken up, which is the bomb sniffing plant idea. So basically, it's a plant that's acting as kind of a sentinel, and then when it interacts with a particle that's found in bombs, and that happens a lot because, they're taking up water, and in the water there are these small particles, and then basically these nanotechnology based particles interact with that particle, and then they change emission profiles, and you can see that with an infrared camera. So if you monitor the plant with an infrared camera, you can see if they're detecting this other particle, which then allows them to sniff bombs, kind of thing.
The only I was going to point out that already there's so many to go into. I know that some of the jet fighters that they've been making, they have the sensors on them where the pilot can literally think something instead of having to use the stick, and the plane will respond to it, which is absolutely amazing. I wanted to say just to Alexander. Yeah, and that's working. That's actually in use. One of the things I want to tell Alexander. So this is my hard drive right here. It's just called pen and paper. It works beautifully, but for one problem, which is it's just a whole bunch of pages, right? And I just open it up and then just start writing, and then I forget where in the book I put it. So I got to write that down somewhere else. But then I forget where I wrote that down. So I've sort of. Yeah, I haven't figured out how to beat that yet. Greg, work on the drive, would you? Would you get the drive thing done? We are now asking you to get the drive thing done. I think that would make our lives just infinitely better. So, what's some of the stuff that you've been working on that you either some of the stuff that you've been working on or the stuff that you find most interesting that they're working on right now? And so I had misspoken. Well, I'd misspoken earlier and was talking about nanobots, and we had some confusion there that you had to clarify. So, so what is it that you're working on right now that you find most interesting?
I guess I think we should kind of set the basis for like what nanoparticles and nanotechnology and kind of like is a little bit. We're kind of like getting deep, but let's like do a 30-foot, 3,000-foot view thing, kind of thing. I think so. Nanoparticles are in the atmosphere and everywhere. They actually existed in the environment before humans. This is a common misconception that it was because of humans and our plastic microparticles and nanoparticles that we've kind of introduced nanoparticles to the environment, which isn't true. So like when you're, for example, like a fire or something like that is actually going to emit multi-walled and single-walled carbon nanotubes. This is my figure. So this is like a carbon nanotube. If you look at the plant cell here on the top, so I actually wanted, this is a publication in a plant journal, so I wanted to put in like a human hair or something that we could kind of like use for reference, but it's a plant journal, so they kind of like, we're like, no, we're not going to do that. But basically the plant, like human hair is also different waves, different widths in the diameter, so it's kind of hard to just say, but human hair ranges between 50 and about 150 microns in diameter. So it's actually about the same size as that plant cell. They're really thin human hair. And that thing in the center there is Chlamydomonas ranhardi, which is really the staple organism that exists for eukaryotes and cell biology. And that's the one that I work on. And then in between the gold microcarrier, so the gold microcarrier is what we usually use right now in order to carry DNA into the chloroplasts of the algae. And in between those, it's actually there, but you can't see it because it's so small. And the zoom in on the bottom half is a bunch of nanoparticles that we use. So mesoporous nanoparticles are actually used quite often because they can actually stuff inside them. Think about them like a wiffle ball. So you can actually put RNA sequences inside of them, and they'll kind of like slough off over time and from the inside the wiffle ball. So this is how, for example, the light emitting plant. Oh, sorry, it's my kids. Sorry. So inside these, some of these nanoparticles, you can actually have RNA transcripts that can deliver RNA scripts for Cas9, which is for CRISPR, stuff like that. Quantum dots or carbon dots, those are about 10 nanometers in diameter, and those can actually be adorned with other things, so you can deliver things with them. But this is basically around the size of like when people talk about those microplastics and stuff like that. Nanoparticles are specifically between the ranges of 1 nanometer and 100 nanometers. So once you get above that, people kind of like say like half a micron as opposed to 500 nanometers. Carbon nanotubes, like I was talking about before, they're about, you can actually order them in different diameters, but 1 nanometer, 5 nanometer diameters, and they're around 100 nanometers in length. And you should think about these nanotubes, and they kind of array inside of an organism, not like a dried noodle, but kind of like as a hair. They actually kind of like, they're electrointerstatic, kind of like puts them together, so they're actually kind of like wavy and loose. It's not like a syringe needle necessarily. So then, like, what kind of scale are we talking about, right? What can we compare these to? So we're on the scale of like proteins now, and you can actually see the tobacco mosaic virus, which is a very popular virus, and it's actually smaller than that. So it's pretty crazy. This is all drawn to scale, by the way. So these are actual. This is all drawn to scale. So it's actually it's really cool. These, a nanoparticle is basically something that's in that 1 to 100 nanometer range. Nanotechnology are these particles that have been functionalized. So the functionalization includes a lot of coatings, a lot of, like, for example, the single-walled carbon nanotubes are actually they're different than multi-walled carbon nanotubes. The multi-walled carbon nanotubes will be like tubes inside of tubes, and you're actually extracting those, and you're just getting single-walled carbon nanotubes, for example. Quantum dots will actually have different coatings on the outside, which will give different properties, different charges. And that's kind of like why these things have such unique properties, is partly because of these weird characteristics that we can give them, and it allows us to do different things and target different things inside of a cell with them. And one of my biggest misconceptions that I had before I went into nanotechnology was that like this was a brand new field, and because I hadn't heard about it, I heard about it in movies. The Iron Man suit, he was like, oh, this is a nanotechnology suit, all that kind of stuff. I'm like, this is brand new. It's like actually like nanotechnology as a field has been going around for like 50 years. It's been studied even more so as in like nanoparticles and as a part of environmental toxicology. So that's kind of like the idea of these things exist, how lethal or toxic are organisms, are these two organisms, how do they impact things, where are they? So the kind of like the monitoring is the environmental toxicology application, but then actually functionalizing the nanoparticles for a specific reason for our designated purpose for research is then nanotechnology.
We have another question from Sinister Porpoise. Is this FRET technology a DNA computing method? And Sinister and I are also both thinking. I know I'm tempted to go down the road, but we'll leave it at that. We're getting into coding biology. I'm going to be a little. I think we had the discussion last time, but at any rate, so that the question is this FRET technology a DNA computing method?
So. Okay, so FRET is when two proteins interact. So they're proteins. Mm-hmm. They're going to be when two proteins interact, they're going to change their wavelength, and in the presence of each other, they're both going to have separate wavelengths. But then when they come together, they're going to completely change into something else that's brand new for an emission. Everything has an excitation and an emission. So you can excite something at like a certain nanometer wavelength, and then you get an emission, at a peak in a spectrogram that you can actually narrow it in and say, I only want to look at this part of the graph. The DNA-based stuff is actually called FISH, so fluorescence in situ hybridization. So this happens when you have like a piece of DNA oligonucleotide. So think about it as like a single strand of DNA, and then with a little kind of like dye or fluorescent marker on the end. So that DNA piece is going to be put into like the cell, and it's only going to emit that DNA, that fluorophore or that dye signal specifically when it binds. So you can detect specific DNA sequences within a cell. And that could be used to make a protein. So the ability to detect proteins and make them based off of the certain wavelengths that are like inside the cells. Think something called optogenetics. So optogenetics, you can actually shine a specific wavelength at an organism and then change the expression of proteins. You can increase expression. Technically, isn't going to be proteins. It's going to be RNA, and then RNA is going to be translated to proteins. But you get the idea. So the presence of proteins is going to happen because of certain wavelengths. So you could theoretically do that. You could have through synthetic biology, you could have a fish probe that then is sets off another FRET signal that then creates another protein, and then that protein then hybridizes with another protein and creates a third signal. And this is all very easily done with confocal microscopy. A lot of what I talk about a lot with my undergrads and stuff is like there's a new way of doing biology in the last 50 years has been in vitro, where we kind of pull the DNA out, we pull the RNA out, we pull the proteins out of the cell, and then kind of do a lot of stuff with them, molecular biology, that kind of stuff. The new wave and where things are kind of going is like in situ. So, or in vivo. In situ is as it is, where it is. So you kind of like go into the cell where it is, see what's going on there without actually disturbing it, without actually like killing everything within it. You're just trying to like see what the DNA is present, what proteins are present, without actually just like killing everything and lysing everything and then taking all the DNA out and then doing some tests on it.
So I did want to ask, how do you, let me rephrase that. How complex can you make the logic gates? I mean, it was for those who are so to clarify, well, computers are basically, ones and zeros. We know this, and then logic gates from the ones and zeros and or so on and so forth. And the computer is really actually very simple. It's nothing more than those components, and then you combine enough of them together, and you can do wild things like screw up your iPhone. But how complex do you think How complex can we get right now with these logic gates, and how complex do you think we could get in the future?
I mean, if it's pretty, like how many nested, you can do like a couple of if else statements pretty readily. You can do, nested, like one nested if else statement. The limitation is really like how many things are you trying to look for? Are there tertiary kind of impacts? Like you're making a product and then the side product that you make is now interacting with it. It's just, it requires you to do more controls and stuff like that. In general, when you're expanding when you're doing an experiment, you're trying to look at all the ways that, the problem is basically like, okay, so now you're doing all this protein stuff and now you're impacting the cell innately. Are you really studying the cell that you wanted to do? If you're making 5 different proteins, what are the off protein or off-chain reactions of a lot of this stuff? So you could do more than we probably want to do right now, just because it is like not exactly 100%. You want clarity in your results, and the dirtier it gets with, all these logic gates and stuff, it can kind of be tough. But I mean, I've seen presentations with synthetic biology professors where you're like, whoa, like this is incredible. I can't believe they're actually doing this at all. I mean, it's incredible. So yeah, it gets pretty gnarly.
So what
would we use this for?
Oh, please, yes.
Synthetic. What would we use this for in the real world?
So what we're talking about, well, I mean, yeah. So this is like, I mean, we're talking about research applications right here, right? This is research, basically. You're not going to be using like complex logic gates and stuff in order to just like make a protein, because we can just make an organism that just makes that protein and just does that really well, right? That's going to be kind of like the goal of a lot of the biotechnology and stuff. But the idea is to use these tools, synthetic biology, in order to study microbiology. Microbiology is basically the last frontier of biology, in my opinion. I'm a microbiology PhD candidate, so I'm a little biased here, but I mean, it is the last frontier. Like if you like cowboy frontier science, if you like next level exploration, if you like, all that kind of like lure of the open ocean and all that kind of stuff, like, I mean, microbiology is where it's at, because 99.9% of microbial species that exist on this earth have never been genetically messed with. We can't even make them in the lab. So they're basically unknown to science, but we know that they're out there. And these 99.9% of microbes have new biomaterials, new biofuels, complex ways to create and mediate protein function, and you're talking about these logic gates, like these new species could have even crazier ways and regulation of making these proteins. So that is a new frontier of science for research to be done. I mean, think about CRISPR. CRISPR is something that's like in like 30 to 40% of all microbes, because they're all basically exposed to viruses. So they're evolutionarily pushed in order to make this stuff. So I mean, if we're missing 30% of CRISPR, 30% of microbes that have this thing, and 99% of microbes we haven't even studied in the field. Like, there is so much out there. It could have huge implications on, like I said, biofuels, materials that we use. It's just food. That's another one. The implications are huge and vast.
It kind of sounds like we're at the point of where we don't even know what kinds of things we could discover. Is that what I'm hearing? It's like there's so much unknown in this field that you have no idea what the future usefulness in the real world would be.
Yeah, I mean, like for example, Jennifer Doudna in Berkeley that discovered CRISPR. She's researching for 20 years before CRISPR was discovered. She was looking into bacterial defense mechanisms from viruses. She had no idea that she was going to be discovering the next generation in genome editing tools. But that's kind of like the point with science, where you just have a hypothesis and there's just something out there to discover, something that's there that we have no idea about, and that is just enough in order to see like what's happening. And what happens is, and this is why I think that nanotechnology actually really ties in with synthetic biology. This is what actually this review that this figure is from. I'm actually making the argument here that microbiology needs nanotechnology as a carrier for the synthetic biology technology. So if you think about like the chassis, kind of like a race car or a boat or whatever, you're like sending it out into these microbial communities, and you want to make sure that they go to a specific cell. You don't want to just like put DNA in there, right? Because then any cell can take it. It's much better to be like, okay, if you're a cell that has these channel proteins in them that I think that are associated with bacteria, I want the bacteria to take this in. I want the yeast to take this in. I want the algae to take this in. And then all these different things have different things for that I want to discover within their chloroplasts or their mitochondria or whatever. So synthetic biology allows us to study what's in these cells without disrupting the entire community. Because the reason why 99.9% of microbes aren't culturable in the lab, they're not grown in the lab, they can't be grown in the lab, is because they have a lot of networks and they rely on one another in order to exist. So once you remove one of them, that the entire community falls. It's kind of like a house of cards. So we need to like go in there kind of stealthily, kind of like without actually disrupting the community, and to kind of like survey kind of secretly like what's happening. And that's a way that nanotechnology serves that purpose, because it's delivering those kind of like ways to figure out what's going on. It can deliver us into those cells specifically. It can deliver those proteins and the RNA. It can deliver the DNA that we need to specific cells.
So is this like, it sounds like applications would be delivering medications, gene therapy. So if there's a genetic component to certain kinds of cancer, maybe trying to edit that out, is that the kind of thing we're talking about?
Yeah, so like nanotechnology would deliver the biomolecules, right? So the nanotechnology is going to deliver the DNA, deliver the RNA, deliver the proteins that have already been made. And then the synthetic biology or whatever can like just be used, to see what's happening, the presence of the presence of the DNA or that's in the cell, the presence of specific RNA, the presence of specific proteins. That's kind of like the surveying kind of thing. There's other applications, right? So this gets us into kind of like the COVID vaccines. Like why were lipid nanoparticles used? Well, it's because, if we just tried to deliver RNA, RNA is going to be eaten up. It's actually very not stable, very unstable. So the lipid nanoparticles kind of like encapsulate the RNA, and they can deliver it into the mammalian cell, which is what we've been doing for 30 years. I mean, we can do that's old technology. That's very old technology. If you go into any nanotechnology lab and you're like, I want to deliver RNA by lipid nanoparticles, they're going to be like, what are you talking about? It'd be like, I want to discover the Model T. Like, I want to do the Model T again, guys. Like, yeah, we've done like, why would you? We did that. We're on to the next thing. So like, for example, and this is all, by the way, all the money and stuff is all in mammals first, obviously, right? Because all the funding is connected to humans. So, we're delivering RNA now with mesoporous nanoparticles. We're delivering them. We're actually putting RNA and adorning carbon nanotubes with RNA now. And these, have specific ways to like get in. And we actually think that there's specific mechanisms that allow these carbon nanotubes, for example, to get into, cell membranes that lipid nanoparticles just can't do. So it's a better efficiency. Higher, more cells are actually taking these up, and they protect the RNA and the cargo. If you think about it as a chassis, they're protecting and delivering at the same time, which is pretty cool.
So is it just me, or did someone suggest that you're trying to deliver COVID vaccines in salad dressing? Did I hear that right? What?
Oh my God. Yeah. So one thing that you can do, so you can basically deliver. Okay. So
just a simple yes or no, sir. I don't understand why you're getting so nervous.
So you can use chloroplasts. Chloroplasts are very unique. They're prokaryotic genetics. So they're basically bacteria genetics inside of a eukaryote. So this is something that I worked on, making like immunotoxins in algae chloroplasts that could kill cancer, non-Hodgkin's lymphoma stage 1, and using that, and then someone else did malaria vaccines inside of chloroplasts. And then the idea was, because of the COVID vaccines, it was, okay, I wonder if we could do mRNA vaccines within chloroplasts of plants. So the idea is to deliver and make these COVID RNA vaccines within plants. And in order to do that, the carrier right now is kind of like carbon nanotubes is a pretty good carrier. There's also gold nanorods that I didn't put in this review figure. I probably should have, but that's just, something that gold nanorods are actually kind of like new. That's like the next kind of like thing that people are kind of studying. But carbon nanotubes have a long history of delivering DNA and RNA and proteins. And so the idea was to deliver the plasmid, make the RNA vaccines, and then make the COVID vaccines, and then that got taken up by like the QAnon conspiracy people to think that we were trying to make COVID vaccines in salads, and then that kind of like got misconstrued by Michael Flynn or something on his podcast, and said that we're actually doing
big shock
in salad dressing. So we were actually putting it in the salad dressing. So, yeah.
Well, to be fair, you said that 99% of these organisms we haven't discovered yet, which sounds to me an awful lot like you're suggesting that Bigfoot is real. All these organisms we haven't discovered, so you seem to think that Bigfoot is so. To each their own. I don't necessarily concur with that opinion, but I'm not going to judge.
There you go.
Totally judge it.
It's true.
Yeah.
I will say this. I will say this. If you get to the point where you think that you found something, like say you're trying to cure cancer or something like that, and you're like, okay, we've got a carrier, we've got all of this. Now we've moved on to human trials. Mm-hmm. If ever you get an applicant named Khan Sunian Singh, do not accept that person into your program. Remember that, Khan Noonien Singh.
I'll write it down right now.
Anybody with that name into your program, it will not end well.
It's definitely not. It's been forecast. So, but as Andrew brought up another point earlier, which I thought was interesting. So I've heard about some of these things that we can make microorganisms that can, for example, eat plastic, and the waste product is something that's beneficial.
Yeah.
How far have we gotten on that, and what does the future look like for that?
That's a good question. I don't deal with that personally. So yeah, there's like nyelinase and these things that
Bigfoot takes up a huge part of your time. I get it, but anyways.
It is a solution possible solution for the plastic waste that's happening right now is to basically use microbes that we found that have an special affinity or ability to actually use plastic and break down plastic. It is a cool thing. Like personally, I'm using the nanotechnology to better algae to make them more the photosynthetic rates higher, hopefully not letting them deal with stress. You can let them able to deal with the higher heat, that kind of stuff, better. And that's kind of like where my personal research is going. But there is definitely a lot of plastic waste out there. Obviously, there's a lot of just waste in general. But yeah, I mean, if in theory you could put out a microbe that does go out and eat just plastic, because the other thing is, the consideration is like, if it's eating just plastic, what else is it going to be eating accidentally? And that's kind of like, that's one of the problems with the biological control mechanism. Like for example, Australia with like their, like, what is it? Rabbits, their goats, they tried to put out this organism that's going to kind of help, but then organisms evolve and they kind of like get out of whack. And there's you have the famous example right now.
That's the beginning of most of your post-apocalyptic sci-fi style movies. Mm-hmm. I mean, that's the entire premise of Jurassic Park franchise. Yeah, nature finds a way.
Yeah, I'm gonna get that clip. We'll put that into the soundboard there. Nature finds a way. I think that's a good one.
There's a really interesting story right now in mosquitoes. So mosquitoes, kind of like famously, ecologists kind of like So there's things called ecosystem services that microbes do or organisms do. Like for example, algae create more than half. If you include cyanobacteria, I mean, algae is like 60 to 70% of the world's oxygen production. So if those go away, how much is it going to cost us in order to make all this oxygen that we need, right? So those are called ecosystem services. There have been papers in ecology that have argued that mosquitoes serve no ecological purpose other than like a driver of evolution, which, I mean, that is, a big part of it. Like our own genome is filled with virus DNA just because of that. The goal of this thing is called a gene drive. So a gene drive is basically the idea that you're going to put two parts together when you make one product. When you make when two mosquitoes make a baby, they basically they can't. It's like they die. So the idea was to use CRISPR as a new like genetically modified kind of like initiation of these gene drives, and it's kind of like gotten a little bit out of control. I don't want to worry anyone, but they did have like a hybrid, that's very odd. That wasn't supposed to be able to be, possible with like part of the CRISPR, and then the other one like mutated with another species. So now it's kind of like has this weird hybrid that they weren't expecting. So it is kind of like an interesting example in nature.
You've just spawned an entirely new line of conspiracy theories right here.
Yeah,
everybody knows
it is true. Yeah, gene drive. It is very cool.
It's gone wild.
It's a very cool thing.
And the mosquito mating thing, you do realize this is a PG show, right? I mean,
sorry.
I appreciate. I don't know your hobbies. I mean, are yours. You don't need to put. But well, no. So I've heard that about we could literally just make mosquitoes not a thing, and why not?
Oh yeah.
So that brings up an interesting.
Please do that.
No problem. After I solve climate change, I'm going to do that one next.
That's a tough call.
I was going to say, I think getting rid of mosquitoes should take precedent over climate change. Well, freaking bastards.
I bet.
I hate mosquitoes with a passion.
Yeah, I'm with you.
So we had discussed on a previous show specifically about science conspiracies and why they exist and why they actually, in some cases, do serve a bit of a purpose. But it seems to me that they exist largely out of a lack of education, but they would still exist even if everybody. I mean, we conspiracies are real, and we do on occasion find real ones, and it's some kind of rigor that offers pushback against what we see, and especially what we don't understand. I think a lot of the scientific conspiracies that we see are, in fact, more of a pushback on the parts of science that people don't understand. They don't understand how it works, so they invent these fear models, and that doesn't mean that science isn't necessarily sometimes abused, or that it doesn't sometimes have consequences. And just because a conspiracy theorist is way off base, doesn't understand it even enough to object to it or understand what they're talking about, that's really an expression of this fear of, what if the scientists, who I can't even really understand what they're doing, aren't kept in check and do something really crazy that has consequences that they can't predict. So I assume that this is a concern of yours. I assume that this is a concern in most labs. So what do scientists, when they're on this exploration and trying to find new technologies, new bacteria, new ways of doing things, what do they do to try to keep unintended consequences from being something really extraordinarily negative?
There's a lot of like internal controls that happen. So like at the university level, where they'll come in and make sure that you're disposing properly, you have the right stuff in equipment in order to store it, you're disposing it right, you're not exposing yourself to it, and then there's kind of like national or federal regulations too. Then this is the BSL kind of stuff. So there's other safeguards that happen. You have just like for example, like my algae. I don't want it to get contaminated with bacteria, because then it kills my experiment. So then I have like a biosafety hood that only allows it to stay there and keep the algae clean. But then also in turn, the algae isn't getting out, but the algae couldn't get out and live anyway. So there's a lot of there's just a lot of the mechanisms that happen within the scientific world. But I would say that most scientists actually aren't really concerned about their work, how they're going to be portrayed as like conspiracies or touching that kind of stuff. Most scientists are kind of like in their worlds, and they it's a field that you really have to devote your life to. And so especially a research scientist that's really into it, that has a lab, or they're the principal investigator, PI as it's normally called. They're teaching, they're applying for grants, and they're doing their research, and they're not really doing much science outreach in general. It does happen, a couple of events maybe a year, but they're not like sitting there, looking up how it's going to impact how it's going to be perceived or something like that. And I think that's a huge problem. The idea that like scientists that aren't being engaged like at City Hall, their local City Hall, to see like, hey, what does City Hall actually need? What kind of research do they need? I think that's actually a huge opportunity for people to actually get involved, like scientists getting involved with their local municipal governments and actually like improving their communities directly. And then on top of that, there's kind of like this thing that's happening where the education level, the science education level, and of even the undergrads, even at undergrad level versus kind of like where research science is, that gap is kind of growing and growing and growing. And it's very frustrating. It's kind of like one of the reasons why I have a YouTube channel, why I do this, is I consider this as, part of my science outreach, where I'm hopefully like informing people of the cool things that are happening, so that people can get stoked that cool stuff are happening and maybe get involved with science themselves, but then also kind of like see that like, dang, they're like, if they're, doing this cool stuff with this new nanotechnology, then maybe we have kind of like lipid nanoparticles figured out a little bit better than you think it is. So then maybe it's a little safer for your vaccine. That's kind of like the big goal for me.
Well, and of course, GMOs. If you've ever, I used to get into I used to debate reasonably frequently, and I'd debate with people like, I will never eat a GMO. I was like, Are you sure you haven't already eaten GMOs? I have never eaten a GMO. Okay, so you've never had rice, wheat, apples, corn. You've never eaten any of those things. Those aren't GMOs. Oh, excuse me, boo boo.
Yeah. I mean, this is an interesting thing. This is an interesting thing because it's like, like genetically modifying something is very specific. So it's a very specific part of the genome that we're taking out or removing or replacing or something like that. And I mean, I'm a microbiology PhD candidate, but I kind of like, cut my teeth on molecular biology and genetics. So that's kind of like what I did for 7 years before going into a PhD program. And to easily tell people like what I did was I basically genetically modify organisms for a living. And I did that with plants and yeast and that kind of stuff. And I mean, when people say the same thing, like, "Oh, I don't like GMOs," and I'm like, "Hey, I killed cancer with an algae through genetically modifying it. So if you know of an algae that does that without genetically modification, like, let me know, please. Let me know so if you find that.
Find one in the wild.
Yeah. So that could be part of the 99%, right? That I was talking about. They really could be, part of it. I think the really good analogy is basically like breeding is kind of like carpet bombing, like kind of a city, and just kind of like leveling everything down. Genetic modification is really like tailored to like kind of like a Navy SEALs kind of like SEALs Team 6 going in, very specific, very like, one building, one person kind of thing. And we have ways, and especially now with sequencing. Sequencing is so cheap. I mean, you can sequence now 800 base pairs through Sanger sequencing for $4. I mean, that is absolutely incredible. 800 base pairs is about the size of, a protein, a small to medium sized protein. So, what you're doing is you're checking where it is and where that landed inside of the genome. And you can do that pretty easily now. So the like, oh man, where is this going to create some crazy hybrid that you don't know about? And it doesn't really happen that often. It's possible, only Siths and non-scientists speak in absolutes, so I can never say it can't, but so it's genetically modifying stuff is very cool.
Well, actually, I want to get into that in a second, but Jake 4D, you can't kill off mosquitoes. Malaria has a right to life. Just ask SCOTUS. Yeah, well, to be fair, listen, I was going to compare Judge Clarence Thomas to a virus, but I have too much respect for dengue to do that. At any rate,
well, this is an interesting topic just because I actually have. So in microbiology, the kind of like, I think it was like 50 years now, they actually changed the living classification to not include viruses. And I don't know, I take it not as like a troll, but like there are active debates happening in science. Science is not settled right now. And I personally, I do think that viruses are living. That is something that I do believe. But gun to my head, if someone's like, "I'm going to kill your kid unless you answer this correctly," like microbiology does not see viruses as living. But just there's actually I debate my biologist friend on my YouTube. There's like a 2-hour debate that we do. I think it's one of my best videos. It's really fun. So. Yeah.
Don't worry. If it comes before the Supreme Court, Clarence Thomas will make sure they're alive, and you have to do what they tell you. So it actually seems like so it's sort of the difference between traditional drug design and rational drug design, where traditionally, basically, the way we would discover that medicine works is we'd try it and see what it does. That would, over time and enough trials, would figure out kind of what works. And then rational drug design, we said, well, let's figure out, what molecules are in play at the target sites of an illness, what medicines might target those sites, and actually take pictures of them, understand what they are, and then figure out what would work just by looking at it, so that we can ahead of time try to figure out what's going to work by examining the actual mechanics of it, instead of just throwing everything we can. The so-called carpet bombing example that you gave. Is that sort of about what we're talking about?
Yeah, I mean, this is like some chemical engineering stuff that's really cool. Proteomics that I don't directly work with, but yeah, you're right. They're through AI and the ability for us to kind of like know secondary and tertiary structures of molecules, we can kind of like understand active sites a little bit better, and we can kind of understand what works and what doesn't work. There is, I'm not going to lie, there's a lot of gray area here. There's a lot of like Rubisco, for example. Rubisco is the hand that brings down the CO2 into the chloroplast for photosynthesis, and CO2 and oxygen get mixed up a lot in Rubisco, and when it grabs O2, it's metabolically not good. When it grabs CO2, photosynthesis happens normally, and it's really good. So that's actually something that we, in the kind of like chloroplast biotechnology space, have kind of like identified as a way to actually improve Rubisco, is by making it more specifically attached to CO2. Like, how do you do that? How do you engineer Rubisco to have a better output and specifically do this? That's kind of like a huge part. There's 12 active sites on Rubisco, and it's not as easy as you'd think. How you do that is kind of like up to your kind of field of science. So like AI, for example, is kind of like looking into, how are other Rubiscos formed and all that kind of stuff and throughout the world, and which ones like kind of like predicting that kind of stuff. I love evolution. I'm a biologist, so I'm actually really interested in using evolution as a way to kind of weed out which mutants are actually going to be more beneficial. So this is called a process called directed evolution. Frances Arnold just got the Nobel Prize, I think it's like 3 or 4 years ago now, based off of directed evolution. And I think she has some incredible papers. She's one of my favorite scientists out right now. So if you're interested in really, really cool chemistry and doing that kind of stuff, like that discovery work, I'd check out Frances Arnold.
It would almost certainly be over my head, but we'll...
Frances Arnold.
No, I meant, are you saying Rubisco? I thought we were talking about crackers.
Rubisco, Rubisco.
Okay, for some reason I kept hearing Nabisco, like the cracker company.
It's true. Yeah, that was it.
Threw me for a loop there. I'm sorry.
No, it's Rubisco.
$6.66 super chat from Dr. Hovind. Can Mr. Review explain why he's trying to turn an elephant into an ear of corn?
Insight. I wish. That'd be cool. There's a lot of people that are actually trying to, theoretically, you can actually... So, like our evolutionary cousins and stuff, like some of their DNA can kind of be in our own DNA. So there's like this idea that you could actually make a mastodon based off of turning on and off genes within, an elephant's DNA, or chickens, for example. You can make a dinosaur out of a chicken, because genes are basically turned on and off. It's called a pseudogene. A pseudogene is something that's basically accumulated mutations, and it's not able to be made anymore. But, when it gets so different, a lot of these things have been turned off, and that's the reason why a chicken's a chicken. Another example of a pseudogene is vitamin C in our own genomes. So humans used to, and most mammals, by the way, dogs and everything, can make vitamin C. It's a crucial protein that's needed in the immune system. So why wouldn't you just be able to make it yourself? Well, we could. The problem is that we were eating so much citrus as apes that basically creating our own, vitamin C was just enzymatically didn't make any sense. If you're thinking about making proteins as some type of like cost part, then, apes that turn that gene off are going to fare better, so that's kind of like why evolution actually pushed the vitamin C to be a pseudogene within humans.
Can we reactivate it? What would be the consequences of just reactivating it?
Yeah, I think that you could theoretically. I mean, that's a target that when people talk about genetically modifying humans, I think that would be a pretty easy one to kind of like a low hanging fruit kind of thing that you probably could.
Are there any other? So, well, yeah, and that's, I think, the kind of thing that does scare people and gets people, I mean, first of all, you wouldn't do this to an unwilling subject. So, people were sitting there thinking that you're just going to start turning things on and off in random people. But that's obviously not how it works. But are there any other interesting dormant or switched off elements of the genome that people have been looking at to maybe turn on and enhance quality of life?
I mean, there's the golden rice example that people have, that where you can, I think it was vitamin A that was put into rice. That got struck down by the European Union, who's very anti-GMO compared to the United States. They're a lot more skeptical. I live in the United States. So I genetically modify organisms for a living. This is how I feel. So.
That was the rice that essentially had a number of essential vitamins in it, so that people in countries where people are not getting sufficient food and they're undernourished, this rice would solve a great deal of that problem. And the EU, which is funny because there's a lot of GMO panic in the US, so it's hard for me to imagine that the EU is worse.
Oh, yeah, it's very bad. If you have a corn, and let's say you insert a gene, and then you take that gene out. So we're talking about 3 different generations, right? The wild type, the mutant, and then the restored wild type. The restored wild type is actually considered genetically modified still in the EU, even though nothing is actually changed. The sequence is completely the same as the wild type, but it's just as a history of genetic modification. So in the EU, it's considered GM.
So I got a question from Jake, and then I think we're going to wrap it up. Well, is entering, do you want to go first, and then?
Oh, well, if you let me go first, I'll put you out of a job, because that was the question I was just about to ask.
Go for it.
So Jake asked a question in the live chat. It says, reactivate vitamin C in the human. Would that have to be done in vitro, or could it be done to a mature adult?
So So basically, the problem with genetically modifying an organism is, how do you genetically modify a eukaryote once it's differentiated? So that's why, I mean, as a molecular biologist, I love working with unicellular organisms, right? Because they're already in single cells, and then you can actually screen millions and millions of them per microliter of culture. So I can do cool things like way more advanced genetic modification than I could with a human. So for me, this is why I prefer, for example, working with green algae as opposed to a land plant. A land plant is going to take 6 to 9 months in order to do this. If at all, if you actually even get it to work, to do one genetic modification. And the problem is basically this exact problem where you get only a certain part of the nuclei that have the integrations, and then how do you select for only those to go forward? And you either are able to deliver it to all of them all at once, which is very hard and difficult and has its own things, and you can do that maybe when they're at really, really low cell counts. So the idea would be to efficiently, maybe through nanotechnology, something like that, because nanotechnology delivery is so much higher than the other methods. You could theoretically do that, but each of the cells might have different have different sequencing or genomes. So that's kind of tough. Theoretically, you could have a nanotechnology vitamin C patch. Okay, so you put it on your shoulder or something, and that creates vitamin C within your cells, and for an extended period of time, kind of thing. I mean, so using the mesoporous nanoparticles, they were actually able to make CRISPR over, I think, is 9 months with an injection of those mesoporous silicone nanoparticles with CRISPR RNA within it. So, these kind of nanotechnology patches or augmentations and stuff could probably be for, an extended period of time, but they're not going to be, permanent because, for example, my skin cells are going to have a different genome than like what's in my gonads, right? Because that they've differentiated, they're not stem cells anymore. And that's why, by the way, that's why you get a sunburn. You get a sunburn because your DNA doesn't match anymore. So your body's literally rejecting that bad DNA now because the DNA has actually been changed because of the sunburn. The UV actually changes the nucleotides in the genome of your skin cells, and your body's literally rejecting it. So it's really cool.
I did not know that. That's okay. Learn something. Wow. All right. So is there anything that you can think of that, so, for people watching this now and who are kind of curious as to what in terms of medical advancements, medications, or dealing with poverty or any other technologies going forward, what are some that you think maybe realistically in the next, say, 2 to 10 years or something, that are some practical applications of this that you think, with the bomb sniffing plants was one? Is there anything else that you can think of that maybe in the next few years will actually be sort of a part of everybody's everyday life?
So it's one, I mean, a lot of this stuff, I mean, humans are so hard to work with, right? This is kind of like why, as a scientist, humans kind of like bore me to actually work with humans. It's really, really hard because the sample size is just so small, and you can't really do anything with them. So it's going to be really tough. So you're going to be looking at like mammals, you're going to be looking at rodents, you're going to be looking at plants, you're looking at microbes. So it's going to be a lot different. I mean, think about it. The lipid nanoparticle delivery of RNA was done 30 years ago, and we're just now doing that with humans. So we're a little behind. Like the coolest nanotechnology, things and papers that I read nowadays. I mean, who knows how long, if ever, that's ever going to be applied to humans. Basically, the coolest stuff that I think about is like, one, selectively delivering specific DNA, RNA, or proteins to specific cells. And that's done with nanotechnology. So David Liu out of Harvard, oh my gosh, this guy's lab is so cool. LIU. That's David R. Liu on Twitter. It's just, it's like an inspiration. I read his papers and I think, I am like literally wasting my time with my PhD. What am I doing with my life? Like, his papers are so cool. They did engineered virus-like particles that basically deliver specific things, DNA, RNA, proteins, to specific cells within the organism. So they did this with, they cured genetic blindness in mice with these engineered virus-like particles. So they had the genetic knockout for a mice for sight. They did the brain scan on the optical lobe or whatever. I think that's right. and then they injected their virus-like nanoparticles that were adorned to specific cells that basically had a localization sequence to those specific cells that would restore sight and restore that genetic knockout that they had done, that the mice were born with. And then they did, again, the brain scan on that specific part of the brain that's associated with seeing, and they actually restored it. They actually restored genetic blindness from these virus-like engineered particles. So stuff like that is just, it is so cool. It really blows my mind. And we're just on the cusp. I mean, personally, I'm really excited about the microbiology stuff, but the ability to actually see and deliver goods and biomolecules, with biomolecules being DNA, RNA, proteins, to specific cells. I mean, that is really cool. Like, imagine you're taking Tylenol orally, and then you're just kind of like hoping it kind of like diffuses through like all the cells in your body. But imagine if it had like a homing tracker that just went right to where it needed to go, right where like that arm molecule, like the arm cells or something were, and all we are is kind of like a collection of cells in our bodies. So how do you find those cells? How do you go and bring and deliver these biomolecules to those specific cells? I mean, that is the cool stuff where you're like, damn, like this is really cool. And then, with humans specifically, you could have, or an organism or something like that, you could have and use a lot of these other technologies to see, if it's working or not, kind of like what we were talking about before with FRET or something like that. So I just see, the world of delivery, with nanotechnology, optics with new microscopes, new ability to see within our organisms. And then, this kind of cool world of molecular biology. I just see it kind of that interdisciplinary intersection right now. Kind of, it's just such a cool place in science. So, I mean, it's a good question. What next? What's next? I don't know. That's a good question. I hope some cool stuff.
Well, I was a big fan of. I'm a huge fan of Aaron Sorkin in general and his writing in general. I think he puts things very well. But there's a scene in The West Wing where they were talking about defunding a particular field of scientific research, and one of the senators involved said, "Well, gee, what is this going to do for us? What are the practical applications?" I said, "None." And I'm like, "Well, wait, why are we spending any money on it then?" I said, "Well, when we started looking at the electron, if you would ask someone what are the practical applications of understanding the electron, none. We didn't know. We had no idea what you could do with an electron. None. It was after we studied it that we were able to invent a world powered by electronics. And, science is the Wild West. It's the Western frontier. We don't know what's out there. Imagine if the Queen of Spain had said, well, we don't know what's out there, so why are we sending any ships? It's exploration is a risky business, and you can lose a lot of money that way, but it's the only way we've gotten anything done.